Okay, yeah, thank you so much for kind introduction. So this is the title. So today, so I just thought what to present. Then, so just to bring some diversity also in the audiences, so that and also the state of the current topics nowadays is COVID-19. So I want to present today some work that our team did on SARS-Coronavirus 2. Basically, we use entropy-based analysis. So just I want to show some quick facts, although everyone is almost updated with these statistics, but still just want to quickly introduce. COVID-19 is an infectious disease caused by novel coronavirus. It's genetically similar to severe acute respiratory syndrome coronavirus, in short SARS-CoV, and it was named as SARS-CoV-2 based on its similarity with SARS-CoV-1 by WHO and declared as pandemic on 11th March this year. So currently, as of today, there are around 26.6 million confirmed cases worldwide, and around 875,000 deaths have occurred across the world. While talking about India, about 4 million confirmed cases so far, and around 69.5 thousand deaths have occurred. So, talking about the genomic mutations in this virus, so the viral genome of SARS coronavirus is about 30 kilobases in length. It's mutating in subtle ways, and exploitation of these tiny mutations can help researchers to track the transition of virus and how this transmission is occurring, and develop viable vaccines and drugs, and avoid an expected second wave of the virus that could also be possible with this kind of analysis. So there are several data repositories where we can find or we can access the genomic data of this virus, thanks to all the researchers across the world. And most popular is GIS-8 and NCBI. They have containing the almost majority of the data that is openly available, and it's a result of collaborative effort among the researchers. So this is just to introduce you about phylogenetic tree. So just before we read the text, it's a tree structure with having a branch node and branches. It starts with a root, which indicates the ancestors, and as the speciation goes on, we have at present, let's say, species of interest are A, B, C, D, E, and there are the present day situation. And so it's a hierarchical diagram that represents the evolutionary relationship between the species, and it maps viral outbreaks geographically and trace transmission chains. So this is the utility of this kind of phylogenetic trees, and study it also helps to study the mutation changes in the SARS-CoV-2. But there are also limitations associated with this. It's complex in nature, and it's not readily quantifiable, and requires complex algorithms and processing to make such kind of trees, and also prone to errors. So coming back to the question that we were interested in. So this picture looks though nice, but it is actually showing us the active cases of the COVID-19 across the globe, and we are here in India is here. All right, so what we wanted to study is the understanding the progression of the COVID-19 in India, and what and how. So so first of all, we used the openly available data sets because of the limited resources. We got data from GIS-8, around 82,000 samples deposited from different countries, and the data is in the FASTA format, and also there is a metadata, means there is also a description of the sample points and date of collection and so on. For reference, genome we took from NCBI with access and ID list for multiple alignment and so forth. So this is the data to show you, and then coming to the methods that we used. So first thing that I want to describe is the informative subtype markers and entropy. So in short, ISM. ISM is a set of positions in the genome of a virus. So for example, you can consider it as its sequence set of positions. It can have T, C, T, C, G, this sequence, let's say. Now, this ISMs, the utility of the ISMs is that it can be used to classify the virus into various subtypes. And another definition is entropy. So entropy, as most of us know, is a measure of amount of information, and high entropy corresponds to high information and can be linked to high mutation at the site. Entropy is used to locate highly mutating sites. More mathematically speaking, entropy is a measure of uncertainty. So this is the formula where we have x as a random variable and hn is the information entropy on the nth site. Xi refers to all the bases at the nth position except n and dash. N is a fully ambiguous site, whereas the dash represents the gap after multiple alignment. So Xi refers to all the, as I said, and Pn represents the frequency of the occurrence of a base out of ACTG at the nth position. Another definition that is relevant from the entropy point of view is null frequency at a position, which is counted as the number of ambiguous sites and this missing gaps after alignment divided by the total number of the sequences. So these are the two definitions that you have to keep in mind, so that you will be able to follow up the next slides. So entropy analysis, basically, as we can say that n and dash are not informative. Then there is another definition called masked entropy, which basically exclude those sequences containing n and dash at a given nucleotide position. So this is basically, now we are trying to improve or enrich the information. So we basically, with the masked entropy concept, we focus on true informative positions. And so the plots that we show in the next slides are the masked entropy of all the nucleotide positions, and it shows the regions of high mutations. So, so there are two criteria that we adopted to take ISMs for final consideration. First is that the positions with the entropy greater than 0.4 were taken, whereas another criteria is that null frequency less than 0.25 were considered. So these are the two criteria to make up one ISMs to be included in our analysis. So we got around 14 distinct positions, which leads to 14 character long ISM. So this is a plot that we got. Here on the vertical axis we have masked entropy, and on the horizontal axis we have positions. So basically, you see for each position you see the masked entropy measurement. So the color coding here represents the different regions of the virus, like ORF1 polyprotein, ORF3A envelope protein. So these are the different functional regions. And so for final entropy, we consider those which have ISM greater than 0.4. So somewhere over here we just put a threshold. So why is so? And yeah, so another thing is that the many points having mass entropy greater than were. So that's why sites with the high n and dash may also have high entropy mass, because they do not represent. They may have high entropy, but they do not actually represent high mutations. So these are like false positives, we can say. So thus we use null frequency to eliminate those sites with having high n and dash. So there are many sites, the null frequency greater than 0.25. Hence, we set a cutoff of 0.25, as you can see here in this histogram on the right side. So we then we having this after having exclusion criteria, we are left with this. Okay, so this is the ISM positions that we got after adopting those filtering criteria. We got around 14 positions for the formation of ISMs. So these are the positions, and these are the masked entropy corresponding to those positions, and this is the location or functional regions that map to these positions. Okay, this is just continuation. So the next thing that we got is to count the ISMs. So how many ISMs, let's say, so these are the ISMs, different ISMs on the horizontal axis. On the vertical axis, we have the count of each of these ISMs. So it's fairly following right side normal distribution. So you can say that hierarchical clustering was also done when we were doing this analysis. So for the in hierarchical clustering, we considered 50 most abundant unique ISMs out of those 568 that we showed in the I showed here in the last slide. So we considered 50 most abundant in terms of frequency, and then we use Hamming distance to calculate the similarities, and also calculate the clustering using the average linkage and UPGMA algorithms. Visualization was done using the dendrogram, or you can say phylogenetic tree, which constructed in the end. So, so this is something which we got from this plot from hierarchical clustering. So as you can see that there are majorly there are 4 groups that I have formed, 1, 2, 3, 4. Well, and the interesting thing is that in 3 of them, and the 3 leftmost of these clusters, we see that China is there. So with the ISMs, we also have included the date and the country of the position of the sequence, you can say. And so these are quite interesting that China is there in these 3 clusters, but it is missing on the 4th cluster. So the reason probably is that in this UK, most of this site in the west, so probably the reason can be that it got highly mutated, that some of the ISMs that were prevalent in the Chinese population where it originated in Wuhan got highly mutated, and that it got disappeared in these countries. So there are further slides to elaborate these things. So this is just a colored mapping of the ISMs, and yeah. So another thing that we checked was to check the geographical distribution of these ISMs across different countries. So what you see here is in this pie chart, you see the percentage of the some of the ISMs that are prevalent. So let's say here in Canada, I just, for example, say that 26% of this ISM was present. So there is also a legend for this ISM here. So if you see that just want to make a comparison, if we look at Canada and Karnataka and Canada, so you see that there is some pattern in terms of kind of what kind of ISMs is prevalent. So you can see there are, except the red one, this is the China. Most probably this is originating from China. This is common, but most of them are really different. Right? Coloring ISMs pattern is very different in these 2 different regions. This is India. This is Canada. All right. So this is showing the same thing, but comparing more countries across the world. So as you can see that that in India, Singapore, and Southern Arabia, USA, United Kingdom, and China. So this kind of ISM that was more prevalent, I guess it was the D type. I will also show some slides later on this D type at 614 position. So this was slowly disappeared when it moved when it go to China to Singapore, sorry. So it goes to 20%, and then it almost disappeared when we go from Singapore to India. So this ISM has so disappeared with the time. And also from UK, as you can see that in UK there are this isotype has grown where with the time, whereas this is it was truly not aligned with the Indian data. Though with the time I will show later the temporal distribution that we will see some more facts about this thing. So if we look at India, so how these ISMs got distributed across the different regions. So if we look at Maharashtra and Telangana, which are now at present in this high prevalent cases are occurring. So these ISMs got kind of similar, but still it's getting the red has gone more prevalent with the time, as you can see here in Tamil Nadu. Whereas in other countries, states like Gujarat and West Bengal, it has almost gone to disappear. So this is the temporal analysis that we did for some of the isotypes. So this is the blue isotype that was in the beginning more prevalent, but with the time you can see that this ISM has almost disappeared, whereas this black one that got highly prevalent with the time. So so this actually but the message here is that why we are seeing such kind of high number of cases in countries like in India, because this has to do something with the kind of mutations that have occurred in the virus and what kind of isotypes now are possibly existing. So this is actually also related to viral infectivity. I think I will show some slides on 614 position. We get some time. So this is the scenario for temporal distribution or for the United States. So for United States, also this was like we had another strain isotype that was prevalent, but then both of them got disappeared, whereas the green one got really highly prevalent. So if you could just look back at India, that we don't see that green isotype at all in Indian population. So another interesting thing, so like in India, the blue one started and the light blue, this is the same isotypes that we saw. So this probably explains that most of Indians are traveling to the UK during that time. So the red one got really more prevalent. So if you look back, so here the black one, so here the green one, and here the red one. So these are different isotypes that are more prevalent in different countries. Well, so just want to spend a few minutes on 614, which is one of the highly region of high interest. So it's a position in the virus genome at position 614, and the mutation from D to G amino acid. So it's a mutation actually in the spike protein. That's why it's so important and so relevant from the point of view that the viral gets more infectivity. So the G variant is more infectious. So as you can see that this kind of mutations has led to infectivity of the virus, and I will show in the next slides that how how this has led to countries like in India and US in worse situations compared to China and other eastern side countries. Why it is more infectious, so that it's a biological reason behind it. So the proteolytic activation of the spike protein is required for coronavirus infectivity, and as this mutation occurs, the proteolytic activation of the spike protein gets enhanced, so that's why it is more infectious. Okay, so if we look at the distribution of the D and the G type, others are ambiguous, and I think the -1 for where there was some ambiguity. Okay, so the interests are D and G basically. So if you see that in India, so this was around 45.6% in the beginning, and whereas in China it was around almost all it started actually with the D type. Then it when as time progressed, the D got converted, got mutated to G type, and nowadays in India we have around more than 50% of the viral are of G type, and also the case in Brazil and USA. So this kind of explains that why these three countries are suffering a lot because of the G type, because it has got more infectivity and it's infecting more people in these countries. Whereas if you look at the eastern countries like China, Japan, and Singapore, the D was more prevalent, and that's why these countries were able to control the virus. Probably there are other reasons as well, but if we look from that point of view, then we can say that that makes some sense. So in India also, if we look at D and G across different regions, so in Maharashtra, that's very interesting actually. So it is still more than 50%. So that may explain that why Maharashtra has got more cases in terms of when we look at the number of cases. So also in Tamil Nadu and in Delhi. So Delhi has done actually also now good job in terms of controlling the spread of the virus. Though still we can see that these are probably some data that explains that the viral infectivity was also higher in Delhi. So if you look at the temporal analysis of so I think I have just one or two more slides. If you look at the temporal analysis that for this position, so D type was really prevalent in the beginning, and as time progressed, the D and G virus are got in equal proportions almost. But the G subtype, or you can say the G isotype subtype of the virus started here around March, end of around March beginning, and then it started getting the more prevalence at the time. Whereas in United States, it's quite similar situation. So it was highly prevalent during the beginning, and then almost around before. I think that also, like, if we I just forgot to mention this, that if you look at back, the G type started around in March beginning, and in USA this started around maybe weeks before. And that's why that also explained that these countries are really got more cases with the time. This is the similar trend that we see in Canada. Whereas if you look at Japan, Japan got highly prevalent only the D type, whereas G type was not able to boost. Okay, so with this I think I am done. So I would like to thank all the team members, especially to Pius and Pratik. They are BTech students and they have done wonderful work. Most of the analysis is done by them. And work. So this is just one work. We are doing a lot of work also in field of computer vision. So ASA is working in that direction. And if you are interested, then